So let's go ahead and get through this third part of the chromosomes, mapping, and meiosis lecture, how the genes are transmitted, the position of the genetic information on the chromosomes, and how that information gets passed to the offspring. So in the last couple of lectures, videos, we were talking about sex-linked traits, information specifically found on the X chromosome, and then we talked about non-disjunction. That's definitely a big thing to remember. Non-disjunction is when the chromosomes do not pull apart. Now, the last part 2, at the end there, I was talking about Down syndrome, trisomy 21. This is where non-disjunction occurs, and the 21st pair of chromosomes is the pair that gets the extra one. So there's actually 3 chromosomes located there. And that leads to our traditional Down syndrome characteristics. We also want to look at other types of non-disjunction. And the main one we want to explore here is when non-disjunction happens on the sex chromosomes. So one possibility is when mom is making an egg and the X chromosome does not separate, it does not pull apart, and 2 of those chromosomes go into the egg. Dad's sperm carries an X, that combines, produces a female, but the problem is the female has 3 sex chromosomes. She's triple X, or also referred to as metafemale. Now, this can also happen if the sperm happen to carry the Y, and that Y chromosome combined with the egg with the 2 X's, and now you have XXY. This is a male, but he has what's known as Klinefelter syndrome. And I'll show you some examples of symptoms and characteristics associated with Klinefelters. It's possible the egg or the sperm, one of those, went through non-disjunction, and it actually lacked the sex chromosome, and that combines with the X from either mom or dad, and you produce a female that has 1 X chromosome and then no second chromosome. So this is what we're looking at with actually a monosomic individual in regards to that particular sex chromosome. This is known as Turner syndrome, and I do have an example of that I can show you guys. If the Y chromosome in the sperm combines with an egg that is missing the X chromosome, so it's a monosomic individual again, then that zygote doesn't even form. It's non-viable. It will not develop. And then the third possible, or the last possible scenario here is if the Y chromosome does not pull apart and the child inherits 2 Y chromosomes. So they were both carried in the sperm, combines with mom's egg, and now you produce a male who has XYY. This is known as Jacob syndrome. So all sorts of different possibilities that can lead to some different challenges for the individual who is trisomic, or in the case of Turner's, in the non-viable, we would consider them monosomic since they are actually missing a piece of information. Okay, so Klinefelters. Here's the general characteristics, features. A lot of times you may not recognize somebody as having Klinefelters, but there are certain physical traits that do develop or are produced because that individual has 2 X chromosomes and 1 Y chromosome. So their body is confused. Do I develop as a woman because I have 2 X's? Do I develop as a man because I have an X and a Y? Which direction does my body go in regards to the development of the physical characteristics? Because keep in mind, with women with 2 X chromosomes, estrogen, progesterone surge and start to get produced during puberty, and that develops female characteristics. The wide hips, the breast development, all those different traits that help us identify here's a female. The Y chromosome, though, is supposed to produce male characteristics. Okay, men are supposed to get broader shoulders, they're supposed to get that beard, they're supposed to get these different features. So the body is just confused. It doesn't know which direction to develop, male versus female. Turner syndrome. Now remember, Turner's is the female who is missing a chromosome. She has one X, but lacks the second. So same challenge here is her body doesn't know how to develop completely. Usually the ovaries are underdeveloped, if there's even really any ovary there. She's got other physical characteristics that indicate she's missing genetic information. Females who have Turner syndrome tend not to have a normal menstruation, if any at all, because her body doesn't have all the information to go through that complete cycle once she's hit puberty. So these are all results of non-disjunction. Individuals who inherited extra information or not enough information based upon a lot of just luck, or unfortunately in these cases, we would consider it bad luck. Okay, so here's the example. Here's how it could look setting up on a Punnett square. Normal male, XY chromosome. Those rip apart during meiosis, and the X goes to the child or the Y goes to the child in the sperm. If non-disjunction happens in mom, two X's stay in this cell. Nothing goes into this cell. If that combines with dad's X, you got your triple X female or metafemale. If that combines with dad's Y, Klinefelters. If that X combines with mom's O, you got Turners. If that Y and mom's O combine, non-viable zygote. So if non-disjunction happens in mom, all four, well, the non-viable one won't develop. Any of the three other possibilities here will have genetic issues associated with them, whether it's metafemale or the triple X, the Klinefelters, or the Turners. So I'm not putting any blame on mom. The same thing can happen in a dad's sperm. As his sperm are developing, he could have non-disjunction in his as well. It tends to be more common that moms have non-disjunction because those germ cells, the cells that will become eggs or sperm, basically are sitting there in the body for all of those years. And over time, they get exposed to things that can create problems with the pathway of meiosis. With males, the sperm are getting recycled so quickly that there's less of a chance. It still can happen. It's just not as likely or as probable in males because of the amount of recycling or basically flushing out the sperm that happens in males. Okay, so the good news, what is good? There's great things going on with genetic testing. So there's all sorts of different ways to detect genetic issues. I would highly encourage you guys to set up your family tree and do what they call the pedigree analysis. Take a look at 3 generations. So you want to look at yourselves, and if you have children, you put your children below you in a generation below you, and then your parents above you. If you do not have children, then you are in the bottom generation, what we call generation 3, the 3rd generation. Your parents, aunts, uncles go to generation 2, and your 4 grandparents go to generation 1. Start looking at just general things, eye color, hair color, height, blood type, widow's peak, non-widow's peak, etc. And you can start filling in the circles or the squares. Circles represent females, squares for males, just to get a feel for how the genetics work in your family. The bigger encouragement is look at health issues. Does anybody have diabetes? There's a genetic link to that. Does anybody have high blood pressure? Does anybody have glaucoma, cancer, etc.? The list goes on and on and on. Those are the things that are more important to look at with your family tree. So the more you can understand the family tree and the potential genetic issues, the smarter we can be with decisions. So if a couple discovers they're pregnant and they look at their pedigree and say, oh, we're kind of worried about, do we have the possibility of passing Huntington's on? Do we have the possibility of passing this on or that on? There are different tests available to detect potential genetic issues. Now, these tests are not normally given. They're given under situations where the doctor may say, well, you're at a high risk for this because we've looked at your family tree, or moms of an older, what they call advanced maternal age. That's usually mid-30s and older. So amniocentesis and chorionic villus sampling are not routine tests every mother gets during pregnancy. It's for those cases where there's a concern that something may be amiss. For some couples, they don't care. I don't care. I'm not worried about it. I'm going to accept my child the way they are. I'm not worried about the test. Other couples may want that information to make different decisions. The decisions are completely the couple's to make. Nobody should be dictating what you do or don't do. But it's a way to give us knowledge and information during a pregnancy. So both types of tests do carry with it a risk of miscarriage. Now, amniocentesis is done usually a bit later in the pregnancy. Most of the time, it's going to be done around the 4th month of the pregnancy when the fetus is very well developed. They draw out some fluid, the amniotic fluid that contains fetal cells. They pull that out with a syringe. So they have to insert the needle through the abdominal wall into the uterus and into the uterine cavity there where the fetus is positioned. And then they look at what's going on with the genetics of that fetus. How many chromosomes? Are there issues on a particular chromosome that you want to discover if your child has? That information then can be made available to the couple. They then can decide for themselves what is the best course of action. If nothing else, they can be better prepared when the baby is born. Now, the chorionic villus sampling is done at a much earlier point of the pregnancy. That's usually done around the 8th week. So we're way back earlier in the pregnancy compared to the amniocentesis, 2 months earlier. Sample of cells from the chorionic villus is drawn out, and the same thing. Let's take a look at the chromosomes of that developing embryo or fetus and do a genetic analysis. And both of these carry risk of potential miscarriage. It is very small, but there is a risk. So they're not to be taken lightly and just, everybody should get one. High-risk pregnancies, pregnancies of concern are when these things are done. So then those chorionic cells are studied, DNA is pulled out, karyotype is set up, and genetic analysis is done. So again, provides information. The couple then can decide what they want to do with that information. 100% their decision. Nobody else should be telling them what to do with it. All right, so when the tests are being conducted, they're looking for things. They're setting up this karyotype. Let's look at the chromosomes. That's what we have over here. Okay, you got 23 chromosomes. Hey, congratulations, you got a baby girl. But unfortunately, she's Down syndrome. She's trisomic. She's got that extra chromosome there. Now, we don't know the expression of the Down syndrome, how much it will express, but based on that karyotype, you know that child is going to be carrying an extra chromosome at the 21st pair. Expression might be very mild, where nobody would have a clue they are genetically Downs, or the expression could go very opposite, 180 degrees the other direction, where that child has severe challenges due to that trisomic nature. Enzymatic function can be detected with one of these samples. Are your lysosomes working? Are they functioning? If not, we're back to that Tay-Sachs disease we talked about. And sadly, your child's going to be born, but they're going to die 3 to 4 years maybe because of that lysosome not functioning. So that's something that can be detected early on in that pregnancy with these types of tests. They can look for diseases located on different alleles. Do you have a gene for a particular genetic disorder? Now, just because we carry the information does not mean it will express. That's the thing I want you guys to remember. It's not a guarantee if you have the gene that it will express. And genetic research is trying to figure out, are there ways where we can either block the expression of negative genes? Or do I go in and somehow put a block on it? Or are there ways we can remove that negative gene, replace it with a positive gene, and change the outlook for that child's life? So lots of potential in the field of genetic engineering. If you guys are interested, there are great opportunities out there for you, especially as a bio major.